and it's my pleasure to interview Dr. Taryn today. Before we start, there's just some house rules. So all phones on silent, please. And for those members online, I see all of you are muted, but please don't forget to mute yourself. And then also for our online members, if you want to pose questions in the chat, please feel free to do so. And Lucky will navigate us through them after the talk. Is it the chat or the Q&A? Chat and Q&A. So you're welcome to do both. And then welcome to our special guest today, the Sparkler Portrait Cat. I'm so excited that you're here. So if ever a talk that you want to see, I think this is it, because it's all about fish movement, fish tracking, which is really, really exciting. So Dr. Taryn Murray, she's my, what you call my science sister in the world of acoustic telemetry. So Taryn has been employed at SAIAB for 7 years, but has been affiliated with SAIAB way before that for 15 years. She started her PhD in 2012, and that's when she was first introduced to acoustic telemetry. And about 6 months prior to that, she started it with Paul Cowley, a supervisor. And 6 months before that, Paul had actually formalized what we call acoustic tracking array network. So we've got these acoustic receivers all along the coastline, and that is now a platform of SAIAB, which is called the ATAP, Acoustic Tracking Array Platform, and it monitors fish movement. So Taryn will tell you all about that. It's really, really exciting. And then, 6 months later, Taryn started her PhD, and pretty much the rest is history after that. She's now manages the ATAP. She took it over from Paul, and her and Matt are doing a magnificent job for all of us. The ATAP acoustic tracking array platform serves the entire marine science community, so people can put in rows to use receivers, can access data. It really is amazing, and Taryn runs this together with that. So Taryn, I'm going to hand over to you. Is it okay?
Perfect. Cool.
Yeah.
What does SAIAB stand for?
So it's the South African Institute for Aquatic Biodiversity. So we study all sorts of things in the water, plants and animals and insects and all sorts of things. Okay. Yeah. Who funds us? we're lots of different people. We get some money from the government, and then we get some money from funny, which is funny money sometimes. We do get some money from other people that we have to basically put an application together, and then we hopefully get some money. So like all sorts of places. Okay. All right. Guys, thank you very much for joining today. Like I said to our more regular faces, this is probably the most nervous I've ever been for a talk because of our special guests over here. But guys, like I said, if you aren't sure, put your hand up and ask questions. Right. We're going to have a cool conversation today. So this talk is specifically focusing on movements between the estuary and the sea for a lot of fish species that we've studied over the last 15 years. It's a very collaborative project, including Amber over here, who did a lot of work on this amazing species called a dusky cob. Because it's all estuary marine connectivity, do you guys know what an estuary is? Okay, yes, yeah. So basically what you've got is you've got seawater coming in, and you've got river water coming down, and where that water mixes, you end up with what's called an estuary. And you've got something here called salinity. Do we know what that is? Amount of salt in the water. Yes, so river is normally naught. The value is naught, and seawater is 35. But estuary is somewhere in between. It can be naught, it can be 35, but it depends on how much water, how much rain there is coming in, and it depends on whether the tide is pushing into the estuary or anything. So these estuaries are actually, you can have a formal definition, which is a semi-closed coastal body of water which is connected to the sea, either permanently or periodically. So that means it is always connected to the sea, or just sometimes. Because sometimes these systems can completely close. Like a big sandbar comes across the mouth and it completely closes for a bit. So yeah, the salinity is different to freshwater and saltwater, and it's somewhere in between. Yes, there are some fish that live in the freshwater and they go all the way down to the sea and they lay their eggs and they come all the way back into freshwater. Fish are super cool. They're really, really cool. So estuaries in South Africa, we've got about 290 of these estuaries, and then we get 42 micro estuaries, which are like really small systems, and they are so varied. You've got really turbid systems like the Orange River on the cool west coast, and then you've got really clear water systems like Cozy Bay, which comes in on the east coast. So as you can see, lots of different shapes, lots of different sizes. We've got what you call these estuary lagoons like Lungaban. You've got estuary lakes. No, what are they called? Estuarine. Yeah, like Nasna, they're something different. And then you get these permanently open systems like the Breede, the Keerworms, the Sundays, the Bushmans. So huge variety in estuaries in South Africa. Now, estuaries have so many different plants and animals and insects and birds and everything living in them. But if we're thinking just about the fish, there are 150 different kinds of fish that live in estuaries in South Africa. And of these, 50 of those are only found in countries that are found in Southern Africa. So that's like Namibia, South Africa, Mozambique. And of those 50, 20 are only found in South Africa, and often just one or two estuaries. Things like your estuarine pipefish or your Nasna seahorse, they may be only found in 2 estuaries in South Africa. Now, estuaries also only make up less than 2% of South Africa as a whole, but they actually bring in a huge amount of money, more than 4 billion rand every single year. So they're like really, really important systems, not only for the fish and the animals that live in them, but also how much money they generate. So estuaries are these super amazing systems, but why are they so important? Well, they have lots of food in them, especially for fish that live in them. They have lots of different shelters and habitats that fish can kind of hide from predators and refuge from predators. And a lot of the species that live in these systems are estuary-associated species. That means they kind of need estuaries in some way. They might live there their whole lives, but they might also just go there sometimes and then go away again. So the stereotypical life history of an estuary-associated fish: the adults go out to sea, that's where they lay their eggs. The little larvae and the juveniles then come back into the estuaries, where they stay and live for 1 to 3 years. And as they get bigger and bigger, they then start heading back out to sea, and the whole life cycle begins again. So you've got this cycle that's ongoing all the time. So why is it actually important to understand why fish are moving between estuaries and the sea? Well, first of all, estuaries are really, really impacted by people. We do a lot of horrible things to estuaries, but everybody likes a beach house, so I don't blame them. So you've got lots of habitat alteration because there are things like houses going up. What also impacts the systems heavily is overfishing. So guys are keeping too many fish, catching too many fish, and keeping too many fish. That's also a major, major problem. Then what actually happened in 2001, which is way before you guys were born, is that there actually used to be vehicles like buckies that were allowed to go on the beach. And then the government said, no, you can't do this anymore. So all the buckies went off the beach. But what that did was take all those guys that were fishing along the beach and basically put them on the estuaries, because fishermen are quite lazy. They don't really want to walk that far. So then suddenly the estuaries were getting fished by all these people. So it ended up there were just too many fish coming out the system, and there still are too many fish coming out. I've included this picture. This is a cool species called the green sawfish. So South Africa used to have one of these. This is actually one of those species. But what happened over time is that they got completely fished out. This fish is now extinct. We don't get it in South Africa anymore. So this kind of thing can happen to a lot of our other species as well. And a lot of our species that are found in estuaries, like these are 4 examples of fish that are really, really targeted by fishermen. They want to catch these fish, are really bad. When it says 2%, that means that of all the adults, all the grown-up fish that can lay eggs, there's only 2% left. So if there were 100 fish, they're now 2. So some of the animals are in really, really bad state. But it's important to understand connectivity because you can answer all sorts of questions. How important are estuaries to fish species? You can understand a bit more about the populations. Are they doing the same thing in different areas? The role of nurseries. When I mean nurseries, that's where the babies and the little fish can grow up. The impact of all these different threats. You know, if you change the way, or you know, add more houses along the estuary, how is it going to affect those fish? And then lastly, and most definitely not least, management. So in order to try and understand in order to develop better rules for the fish, for the fishermen to follow, we need to understand movement. So management is really, really important for what we do. So over the last 20 years, actually it's a bit more than 20 years now, a whole bunch of people have been studying the movements of estuary fish in South Africa, and it was mostly led by these two guys over here. We've got Paul Cowley, and then a Norwegian collaborator, so a guy from Norway, Dr. Tu Nasha, and a lot of this work was done in collaboration with a Norwegian agency. So we've got funding, and the two of us, so somebody was asking about funding, there we go, from Norway. We got money from Norway, and we did all this work together on a whole bunch of different species. So another crash course. What I'm talking about is acoustic telemetry. So in order to study the movements of these animals, where they're going, we use something called acoustic telemetry. And what this basically is you've got these things called receivers that you put in the river or the sea or wherever you want to study the fish. You've got fish swimming around. You go out and you catch them with rod and line, which is really always exciting. You then put a little tag in them. So you actually do a little operation and you put that tag in it. And that tag is then sending out a sound signal. And when that fish is in the detection range of a receiver, close to it, that receiver picks up that sound. And you get the unique ID of the fish, the date, and the time. So suddenly, if you start putting together all these little pieces of of movement, you start building a wonderful movement puzzle. Yeah. Yeah, you can actually, yeah. So sometimes, because nobody knows that these tags are in them, because what happens is you do the little operation and that star heals so nicely, you will never even know that it had a tag in it. So sometimes there is a chance that we can tag a fish again. Yeah. It can be any fish, but the ones that we have an interest in are fish that fishermen like to catch. So they're the ones that are maybe in a little bit more trouble than some of the other ones. Sometimes it's just a really cool fish. Go and study it. I really like catching this fish. Let's put a tag in it. But we mostly do it because we need to try and find some good management. Oh, those are just to look pretty at, yeah. Those are freshwater fish. So those are ones called cichlids. But it's basically because we used to be a fish institute. So this is where we study fish stuff. Always nice seeing fish as you walk in. And they're nice and pretty as well. Okay, so you've got all these pieces of equipment, and together we've actually made this huge nationwide network called the Acoustic Tracking Array Platform. So every little dot that you see over here is an individual receiver. So in South Africa at the moment, we've got over 320 receivers in the water at the moment. And this is covering 2,200 kilometers of coastline. So down the Berg Estuary, all the way on the west coast, up to the South Africa-Mozambique border, a little bit higher in Santa Maria. And at the moment, we're tracking the movements of 900 animals. So this is fishes, sharks, rays, skates, and even turtles. We don't, but our friends at the aquariums do. Because you get lots of turtles that wash up on the beach. So they look after those turtles, they make sure they're big and strong, and then they put a tag on them and let them go. But that's like new stuff. So that's like new work that's starting up. So over the past 20 years, we've tagged all these different species, 14 different species in various different estuaries. So all the ones highlighted in yellow are where we've actually tagged animals. So again, that's from the Berg on the west coast all the way to the Mtentu, which is along the Wild Coast. This has actually produced more than 16 million individual data points. So it's a lot and a lot and a lot of information. So this is just pictures because I knew you guys would be here, and I'm going to put in some pictures of what some of the animals look like. So we've got dusty cob in the top left. We've got a blue stingray, giant kingfish. We've got a couple of mullet species. Bantony, our PhD student, is doing that at the moment. A diamond ray, a duckbill ray, white steenbras, and a leervis. So these are just 8 of the 14 species that have been tagged. So what do we mean when I say connectivity, guys? So this is like movements between the sea and the estuary. So an estuary, you can either try and understand connectivity, like movements in an estuary. So are they spending more time in the upper reaches or the lower reaches or the middle reaches? Or, and for the purpose of this talk, what I'm talking about is estuary sea trips. So it's movements in and out of the estuary or between the estuary and the river. But for this talk, it's only the movements between the estuary and the sea. And then you can also start recording things like temperature. You can put little loggers in the estuary and you get information on temperature. You can look at rainfall or day and night and really start understanding how these animals are moving and when they're moving in and out. So just a couple of case studies to sort of highlight the work that's been done. So this is a fish called spotted grunter, and they've been tagged in a whole bunch of estuaries along the South African coastline. And the big colorful dots don't mean much to anybody, but it's basically like a clock. Okay, so you've got midnight at the top, so that's in the middle of the night, 6 o'clock in the morning, midday, 6 o'clock at night. And each of these dots is the time that the fish either left an estuary or came back into an estuary. So you can see there's just lots of color everywhere. But if you start really looking at each estuary separately, you can see that fish tagged in the Khoko, for example, which is down over here, we're leaving and coming back at very different times compared to the Sundays estuary, which is over here. So some of them, so it's important to tag animals, the same species, in lots of different estuaries because they do different things. If we look at giant kingfish, which is this beautiful fish over here, they were tagged in Tentu, which is up on the wild coast. This study looked at different sizes of animals. So we had really small little juveniles, we had sub-adults, these are basically your teenagers, and then you had your adults. And what we found was that the juveniles were moving up and down an estuary, but they stayed there all the time. Every gray strip over here is winter. Okay, so the white is summer, gray is winter. And for the little ones, they stayed there the whole time. But for the bigger ones, as they got bigger, they decided, hey, winter's too cold. I'm not staying here anymore. And they left. So that's why we keep seeing these huge gaps of the fish not being there. The blue, which is really interesting, is fish, big adults, that went all the way to Mozambique and came back. So these guys move a long way, but they keep coming back to an area that they know and that they're familiar with. So it really highlights the need to sort of study the same species, but different sizes of the animals. This is just a picture of that giant aggregation in southern Mozambique. So it's huge, it's amazing. Biggest one in the world. So just across South African border, biggest aggregation of giant kingfish in the entire world. If we look at white steenbras over here, they call these guys the steam trains. You know what I call them, steam trains? Okay. Okay, so if a guy goes and he fishes them, if a steenbras picks up that bait, it just goes like a steam train. It doesn't like pull, it just goes for it. And your line goes like the whole time. So steam trains. And what we found was these guys were actually tagged near an estuary, so not in an estuary, just next to an estuary. And the adults of the species, they don't like estuaries. They don't go into estuaries at all. But what we actually found was that these big adults that we tagged were going in and out of an estuary all the time. And if you look at these circles, that's new moon and full moon, which is spring tide. So that's when you get really big tides or really low tides. And what these guys were doing is on the really big tides, the spring tides, they kept going into the estuary. So this was really cool because we didn't know that they did this. So this is highlighting how tagging animals near estuaries can also actually answer questions that you didn't even know you wanted answered. Okay, if we look at leervis, this is my PhD species, so I'm very fond of these guys. But if you want to talk about fish moving all over the place or moving to different estuaries, these guys, these are the winners. They're the champions. So we tagged fish in the Kawi estuary over here and down the Koko estuary. And what we actually found is that they just decided to move wherever they wanted to go. So the fish in the Koko were jumping all over the place, from the Breda all the way to the Nasda, where the guys in the Kawi were jumping from the Klop all the way to the Chalamna. And some of these fish actually moved about 500 kilometers away to the KZN coast. So really, really big movers. And again, it highlights the need to put receivers in lots of different estuaries, because if we didn't have that, we wouldn't even have known that they moved. Okay, with moving away from the fish, we're going to the stingrays now. These guys were tagged down in the Breda estuary. You can't see it over here, but there's my map. In the Breda estuary, along with a whole bunch of different fish species, even bull sharks, because bull sharks like estuaries. So there were a whole bunch of different animals tagged in this estuary. And what we found was that these stingrays, especially the ones that were tagged in the estuary, liked coming back to the estuary year and year and years later. So this guy is a diamond ray, came back 6 years in a row. Didn't like winter, so left again, but came back in spring, summer, and autumn, and then left again for 6 years. And what's interesting is that maybe the guys tagged in at sea sort of did the same thing, but not nearly like the estuary guys do. So again, it highlights the need to tag animals at the sea as well as in the estuary. This example has got absolutely nothing to do with movement. So forget about your movement. This has got nothing to do with that. But one of the things we found with tagging all these animals is that some of them, actually a lot more than we realize, are actually caught by fishermen again. How do you think they know there's a tag in the fish?
Yeah.
Exactly. So what happens when they keep that fish and they decide to eat that fish? Yeah. Eat something crunchy, yeah. Yeah, that would be great, because then you can find out where those people are. No, so what actually happens is that when you go and they gut the fish, okay, so they keep the fish, they're going to eat it now, but you don't like eating like all the stuff on the inside, so gut the fish. But once they do that, what is this black thing? Like, what is this? And we stick very bright yellow tags over the transmitters, so over the tags, with a big reward on them to try and get them back, because they cost a lot of money and we can reuse them again. So what happens is that when they gut the fish, they see this and they're like, what is this? And then they phone us and they say, hey, we found something. And then we find out a bit more information. So we actually finding out how the levels of fishing pressure that these fish are facing, like that leervis, the one that like moved all over the place, of that species, close on 40% of all the fish that we've tagged of that species have been recaptured. And you think there must be millions of fish swimming in the sea. 40% of our tagged fish have been recaught. So it's crazy. So anyway, here's a dusky cob. This is Amber's PhD species. So also a very, very special species. And a couple of these, I've typed this dead fish swimming just to highlight how much they recaptured. This is the scale. This is how big they can get. So the scale of amber tiles over here, just so you know how big they are. And there were fish that were tagged in the Sundays estuary over here and fish tagged in the Breda over here. These two estuaries are very, very, very important for this fish species. They're basically what we call the two last strongholds. So pretty much if these estuaries, if something happens to the estuaries, this species is in serious trouble. So we have to really look after the estuaries as well as these species. So 65 were tagged in the Sundays and 10 were tagged in the Breda, all juvenile fish. So these are like small little guys. And what happened was that 33% of the Sundays fish and all the fish in the Breda were recaught and killed. So they've like completely taken out the system. So it's just showing you how much guys are actually fishing for these species. They're actually endangered. So I don't know if you guys know about the IUCN Red List. So it pretty much lists an animal. You plug in a whole bunch of information, like where the animal's found, what kind of threats they face. So is it overfishing? Are they sold overseas or whatever it is? And you come up with this scale on how endangered an animal is. And dusky cob are listed as endangered. And there's been a huge move to try and protect them along with the rhino. So everybody knows rhinos, we love rhinos, but you can see rhinos. You can't see these guys. So you need to start thinking about big fish like this as rhinos of the sea. Yep. exactly. So that's why you have to try and keep. You can catch your fish, but maybe don't keep it. Maybe think about letting it go again. Yeah. Yes. So everybody that fishes, everybody that catches fish needs to have a license. They're not expensive. They're like $100 now, 100 Rand for the year. And then there are rules that you have to follow. So those rules will tell you how many fish you can keep and what size they need to be. This guy has to be over 60 centimeters, but you can only keep one bigger than a meter. So yeah, but landing a meter or even catching a meter long cob is quite, there are not many of them left. Yeah, they all breed at sea. So we actually don't know where they breed a lot of the time, but it's maybe better because then nobody knows who they are. They're just doing their thing. Yeah. Oh, that's over here. Okay, so this fish, there were 60 fish tagged in the breeder, but 50 of those were big ones. I'm talking the smallest was 1.2 meters, the biggest was 1.6 meters. So the 10 is just the little guys, the little juveniles. Okay, so why do we even need to know this information? What's the importance of it? First of all, we can start understanding the influence that all these different, what we call anthropogenic changes. Anthropogenic means man-made. It's what we're doing. So we can start figuring out the influence of these different changes on estuaries, which basically can affect the way that fish are moving. And also, we've got climate change. You guys will have learned about climate change and heard about it. So what's happening is we've got some increases in water temperature on the East Coast. We've got decreases in water temperature on the West Coast. So some places are getting colder, some places are getting hotter. And what's happening, what this does is that some of the fish that don't like hot water, they'll come sort of, okay, so fish that are kind of found here with cooling and heating, they're pretty much only going to be found here. So we've got some really interesting things to understand. The movement can help you kind of track these things. And climate change is also bringing about stronger currents. We've got increases in sea level and then lots and lots of storms as well. So all of these things are going to impact how fish move in and out of estuaries. So an example to highlight this is if we look at rough seas. Okay, where are you guys from? Coast Victoria. Nowhere near this. Oh, that's why you guys are flip checkers. Okay, all right. So what's happening is every now and again, we had one recently, like this huge storm event. I don't know if you guys heard about it on the news, maybe you guys talking about it. But Cape Town, here in the Eastern Cape, we were hit with huge storms. And even down in PE, the sea dumped huge rocks on the road, like water moved giant rocks onto the road. So there's huge storm events that are happening at the moment. And what happened between 2008 and 2009, so also a very long time ago, is that there were these huge low pressure cells that came through, which is basically the sea turned on its head with massive waves, big strong winds, and it's kind of linked to these low pressure systems. So there's a bottom there and there, so very low systems. And what happened is that we actually had animals tagged in 3 estuaries, so 3 different species, 3 estuaries. They're doing their own thing completely. What happened? We had steenbras tagged in a predominantly closed or open estuary, the East Canamonda. And what happened with this rough sea? The mouth broke open. So the fish that were tagged there went out to sea. Well, actually, I'll tell you what the fish did there. Then with the next one, we had spotted grunter that were tagged in a couple of estuaries. And then dusky cob were tagged, I think, in the Sundays as well. So with these 3 huge systems, we had major overtopping. So pretty much the estuary broke out, the sea came in, it was chaos. We had a huge low pressure over South Africa. And this, when you see wave height like this, if it's red and orange, I mean, if it's pink and purple, that's bad. That's like really, really strong. So the sea was so high. And what pretty much happened with all those fish in the different estuaries was that they either left. They said, I'm out, I can't do this, I'm going. Or they changed their movements, or they actually lived somewhere else in the estuary and they decided to shift it. So if they lived in the upper reaches, they moved down to the lower reaches. So what happened when everything went back to normal? The fish did the normal thing again. So they had huge changes in movement behavior. So kind of following what the fish are doing in the estuaries can tell you more about how these huge storms are going to impact on the fish in the future. Okay, yeah, that's it. This is a beautiful picture, I think, of the Karika estuary, which is just down the coast nearby us. So thank you very much for listening, guys. Thank you for being so engaging.
Are there any more questions? What
Okay, so it's actually quite interesting. So a lot of these fish that I'm talking about, guys, they have to go to sea in order to lay their eggs. But what Pumza is asking, what happens if that's closed and they can't go out the estuary? So in that case, the fish just don't breed. They just don't lay their eggs at all because they can't do that in the estuary. That's not an option for them. So they generally don't breed. Even if they are breeding age, they're just like, no, I'm not going to do it. But once that mouth breaks open, they all just bolt out to sea. But it's also, they can survive in an estuary that's closed for a long time, given the salinity and the temperature doesn't go up too much. Because if you get hypersaline situations, the fish can't cope and then they'll end up dying. So we've actually had a couple of fish kills in some of the estuaries where was oxygen, salinity, and temperature, like all sorts of things just resulted in the fish dying. But these guys, if the estuary closes like for 2 years, they should be fine, given some fresh water still coming in. Yeah, so not in South Africa. It's an excellent question. So in South Africa, we don't do, they call that restocking programs. In Australia, they are flat out restocking, especially on some of the very popular fishing species. There's pros and cons to it. This is my personal opinion, so it's not of the Institute or anything, but my personal opinion is that you start running into risk of potential, especially if they're bred in sort of enclosed areas and then you release them, there's potential for sort of contaminants to get out into the system, and you'll essentially start kind of diluting the genetic diversity, because if you've got, say, 10 adults that you're breeding from all the time, at sea they might have 1,000 adults that they can kind of choose from. So they're definitely pros and cons to it, but the Australians are very much on board, especially with like barramundi, and I think they're doing Malaware as well, yeah, which is dusky cod. Yeah. Mm-hmm. They definitely have a ability to learn. So they're smart. The fish are smarter than people give them credit for. So they don't kind of have the same brain makeup as animals or people, but they definitely learn certain behaviors. So if you've got, especially in the lab, we can see this if we can do like experiments in the lab and you put them in little fish tanks, they can maybe learn certain things will give them food, or you know, if you do, I don't know, add hot water. I'm trying to think of really dumb examples, but they'll realize that doing this thing will give us food or something. And they learn quite quickly. So yes.
I'm going to quickly jump to the online platform. So we've got a message from Taryn. Great talk, very informative.
That's my twin sister. Thank you.
Are there systems that enable tracking of the temperature as the fish moves?
Okay. Yes, Roger. Thanks, Roger. So there are actually some tags now that allow you to record the position of the fish along with temperature and salinity. We don't use them at the moment just because of how expensive they are. So we sort of go the option of putting salinity or temperature loggers in the estuaries or the sea or whatever, and trying to link that up with where the fish is. But first prize, and what we've always wanted to do, is look at like temperature and depth tags, because it would just add another layer of what we already know about the movements. Yeah. Yes. Okay. So it depends on the size. They come in a whole bunch of different sizes. I should have actually brought some down, but they're making really small tags now. They're basically just bigger than a centimeter, and the biggest ones are 6 odd centimeters. The small ones will have a battery life of 2 to 3 months. The big ones can go for 10 years, which is why we try and recoup our tags if a fish is caught, say, after 2 years after tagging. Once the battery goes dead, it's dead to any kind of tracking. So unfortunately, if the tag does expire, there's no way of getting it back. Yeah, so that's always the fun part. We've spent many years digging around in very not dirty as in like pollution dirty, but dark, turbid estuaries trying to find receivers. Because unfortunately, well, fortunately, because it gets us out the office, but unfortunately, you have to physically go, take it out the water, download it, put it back in the water. So you get some really cool satellite tags. Those are basically on animals that come out to the surface. So like your sharks, your turtles, big predatory fish that break the surface, like giant kingfish, yellowtail. They can all be tagged with satellite tags. And when they break the surface of the water, that tag relays information to the satellite. And you can kind of get the information either immediately or it stores it on the tag. But these guys, we actually have to go out and get that. Yes, yeah. Oh, and then at sea, we've got the same piece of equipment, but it's just got a release at the bottom. So at sea, you just plug a little code in and your whole unit comes back up to the surface. Yeah, so in the estuaries, generally once a year. So at the moment, we've got a dedicated study in the Cowie estuary, which is in Port Alfred, just down the drag. And we are trying to download those receivers every 3 months just to secure that information because we've got student projects at the moment, currently on the go. But in the estuaries, once a year, at sea, 6 to 8 months. Yeah. Yeah. They generally stay there. We just have to wait a couple of years for the sand to shift again. Hey, receiver. Yeah. Yeah, that's why we have to reduce the time. Yeah. Okay, so the receivers, are you talking about the receivers? Batteries, everything's battery operated. It would be great if they were a rechargeable unit, but I don't know if they'd be able to make one, not yet. Maybe they'll still come, one that can be rechargeable, but at the moment we have to buy batteries all the time. Once those batteries come out, we've just got to, those big ones. You know, like those big fat ones, a little bit bigger than that. D cells, called D cells. Yeah. Yeah, that's right. Yeah, well, we've definitely got in-house capacity to be able to contribute to something like that. So we've got a team, I think at least 5 of our researchers are marine estuary orientated. So we've definitely got, and between XE department as well, there's definitely capacity to do something like that. I'm sure people would be interested. Yeah, perfect. Thank you. Yeah. Yeah, so with these tags, unfortunately, we only find out after they've killed the fish and gutted the fish, but we put a bright yellow reward tag. I think they're actually white now, but reward sticker on it. And on that, it's got a cell phone number and email address. And even if we can't get the tag back, it's just really nice having that information because then you know that something's happened to the fish. It's not just disappeared because that kind of changes the way you think the fish might have moved. So maybe it just disappeared and you go, maybe it went offshore where the receivers aren't. You're like, but if we know that it was killed, then you go, oh, okay, that's the last time. Yeah. Okay, so we generally try and put them at the bottom. So we're really lucky because a lot of our estuaries are super brown and super muddy. So what happens if you put them in a big cement block down at the bottom, and it's at least under 1 meter of water. So because the river's so dirty, like so muddy, they can't see it. But if they are there, we also write research over our receivers and hope that they don't touch them. We've had 1 or 2 receivers go missing, but generally guys are pretty good about it. I think they're more interested than they want to actually damage their equipment.
Then I can see we don't put them on the trawling ground.
Yeah.
We try and put them in different areas.
Yeah. Now we're getting into that stuff. I'm not too sure, but I think once they've learned that behavior, it never sort of leaves them. So let's use COP, for example. They grow up to 40 years of age. They can be 40 years old. That's how long they can live for. So if they learn something maybe when they were 8, they're going to remember it for 32 years. So I think once that behavior is learned, then they're good to go. So probably pretty good. Not like a goldfish at all, huh? Yeah. So we've actually had this 1 or 2 times. Excellent question. The fish is there, it's swimming up and down, doing its thing, and suddenly it just flatlines. And you're like, what's happened over here? Then you know that the guy's gutted the fish. He hasn't seen it, and he's put it back in the river, but it happens to be next to a receiver. And you go, surely, like it could have been somewhere else. But then that tag is just sending out its signal all the time, and the receiver's just going, cool, there's a fish here, but it's not. The other really cool thing that happens is sometimes another fish, so you get smaller ones like the little mullet, they can get caught, eaten by a predatory fish. So the dusky carp or that leervis, or even a shark. And what happens is the fish is doing its normal thing, changes behavior completely, suddenly starts doing some other kind of weird movement, being eaten by one of those animals. And then you're picking up the predator instead of the prey. So there's some really cool things. And
but that will only last for like 2 or 3 days.
No, it doesn't.
Sometimes not.
I was just going to say, I've recently learned that we usually thought, okay, well, they're going to eat it. It's just going to go through their system. They're just going to poop it out. But apparently the tags can stay in the animal for like a very long time. I'm talking like months, like 6 months. You could be tracking a shark. You're like, oh, cool, this fish has gone to President and back. Meanwhile, it's a shark. So, yeah, really interesting stuff.
And the other thing, you can use a metal detector to go and find that tag. So you can snorkel and use a metal detector and find the tag that's been.
Yeah. Ah, cool. Torpedoes, yeah. Cool. Okay, so the torpedo rays, hope you didn't stand on one because it'll give you an answer. Oh. You love to tell the tale. No, so they give you quite a nasty shock. We haven't tagged any. It's been on the wish list, but it's just how do you tag something that's potentially going to shock you back, because that's like a little bit nerve-wracking. The Canadians have actually managed to do it, and they've ended up externally tagging. So they got the little tag that we use, but they just put it in a special case and they kind of just stick it in the ray and hope for the best. Something we might play around with, but it's going to be a real wish list species because it's an expensive game we play, unfortunately. Each tag's 6,500 Rand, so we can't afford to just.
And the ones with temperature and things like that, they're not.
Yeah, they're way more. But yeah, so the torpedo rays are fun, and they fill up in the estuaries. They really love the estuaries. So on our wish list. Oh yeah. My goodness, you're braver than me. I wouldn't touch that thing. Yeah. as exciting as we think studying fish movement is, unfortunately, not many other people find it as exciting. So we haven't had our research done in just yet. But what can happen is that some researchers say, I'm going to study this species. This is mine. You cannot touch this species. And you're going, well, why? It's not yours. It doesn't belong to you. So sometimes we can run into problems like that. But with this community, all of us work really, really nicely together. All of us are friends, which is great, but no stealing data just yet. I'm not going to say it's not going to happen, but it actually does happen a lot of the time in the first world countries like America and whatever. A lot of their data is online. So people can just go and I'm going to download this, and they can write it up, even if it belongs to somebody else. So there are some things, but in South Africa, we're a bit slow on those things. So we're still good. We're still friends.
And also because it's important because we're all working to a common goal. So we all want to conserve fish and conserve oceans. So it's no point fighting against each other because there's so much work that still needs to be done. And if we all work together, then we can achieve it together. We won't even achieve it in our life. It's so much to do.
Yeah, no, this community is really, really nice because it's so small. They're probably only off the top, if I had to put a number to it, like 15 to 20 key telemetry people in the country. Not to say that there aren't other people that are involved, but telemetry researchers. So we're a very small community, and because we're so small, we have to play nice. There's no other option. So that big network that I showed you guys, like all the little dots, those actually don't all belong to SAIAB. They belong to lots of different people, like Amber and her team at Rhodes University. They've got their own receivers. The other people down in Cape Town, Shark Spotters, they've got their own receivers, but all of them play nice. And what we do is collect all the data. So we have it all, and we collect it all and maintain it for everybody. So we manage it for them.
And do you want to start?
We don't, but somebody had interest in it. We don't get them here, so they're only warm water, so higher up on the east coast, so like Ismagaliso, Mozambique, exactly. But the guys in Australia have done lots of sea snake stuff. They do very cool things. And I was actually chatting to a guy in the United Arab Emirates, like higher up, and they were interested in tagging sea snakes as well. How much time do you have? No, because the fish that we tag are mostly the fishermen really like to catch it. So those are the ones that are really interesting. But like the cob, I mean, the fact that you can catch a fish that's this big, it's amazing. Giant kingfish, that other one that goes up Mozambique, huge sport fish as well. They can get up to like 30, 40 kilos. Big, big, big fish. We're tagging a fish from a family called sparrows, and they get really old. So some of them can grow up to like 40, 45 years. So we're getting like big fish. And then we don't do it, but there are a lot of sharks. So a lot of guys are tagging white sharks. We've got bull sharks, tiger sharks, and then the turtles that the aquariums are doing. So yeah, lots of different cool things. If you have ideas, let us know. Maybe we'll start tagging those too. Other questions? Yes. Not moray eels, no, not yet. Some guys have tagged freshwater eels, like Colin, so some of them have tagged that, but they've got a nasty habit of getting rid of the tag. They just feel it in their body and they go, I don't like this anymore, and they'll just like push it out their body. So they do really weird things, but we still want to try and tag them, see what happens. Sounds like you should be a scientist. Do that. No, again, I guess it depends where your interest is. So if you've got people that quite like the biological world, so they're interested in animals and plants, maybe they'll find fish really interesting. You know how I ended up here? By pure chance. I came to a university and I knew this much about fish. And now, I don't know, how long have we been doing this? 17 years later, I'm still here with fish. And I love them. They're amazing.
Okay, so we're going to wrap it up for the online community, but you guys are welcome to stay and answer more questions. Okay,
Thanks for
questions. So thank you to everyone who attended online. I hope you really enjoyed it as much as I did and the rest of us here at SAIAB. Yeah, thanks so much. Do you have any more questions?
You're all there.
You go back to work. But